you have to intervene before the hypometabolism, and therefore you have to make sure that those areas that are going to be the first one revealing that change can be rescued.
Welcome to the Evolving Past Alzheimer's podcast, where we are focused on bringing you information to help prevent from developing and improve from suffering with brain diseases like Alzheimer's and other dementias. I'm Dr. Nate Bergman, a physician and Chief Scientific Wellness Officer at Kemper Cognitive Wellness, and I'll be your guide on these sound waves. So whether you're a baby boomer who's worried that your brain wiring just isn't working like it once was, or you have a loved one with the disease, or you yourself have already been diagnosed and are wondering, what do I do now? You'll want to listen to this podcast. Each episode, we introduce you to the top doers and thinkers that are revisioning Alzheimer's, dementia, and just generally things in life's second half. If you have questions or comments, check us out on social media. To support us, go to patreon.com/evolvingpast and consider hitting the button and becoming a patron of this show if you find these episodes valuable. That's Patreon, Patreon, patreon.com/evolvingpast. So here we go. Let's get better. Hi, everybody. Welcome back. Because of the super technical nature of the interview you're about to hear, and because of the listener feedback we've gotten that some episodes get really technical and we tend to lose people, I want to do a little summary for a few minutes of the information that you're about to hear. Our guest is an amazing, really a world leader in brain energy and its impact on cognition and cognitive change that lead to Alzheimer's. His name is Dr. Francisco Gonzalez-Lima at the University of Texas in Austin. For this first part, we had to break this episode up into two parts because it was so long and so technical. But at the beginning of this episode, we really begin to suss out the difference between early onset Alzheimer's, that's more typically genetically driven, and then the more common so-called geriatric, senile, or later onset Alzheimer's disease that usually happens after age 65. And even though we don't think of 65 as being geriatric anymore, that's just sort of the convention because it's been a few decades. And in some settings, and some neurologists have been using that term for much, much longer. Regarding the typical onset, the later onset Alzheimer's that happens after age 65, Dr. Gonzalez-Lima argues that there's a common pathway. The common pathway is a problem with bioenergetics, or the way that the brain produces energy. So he characterizes the fundamental problem of Alzheimer's being that the brain does not use oxygen to produce energy, or ATP. ATP is sort of an energy cache that's made in the mitochondria of our cells in our brain and the rest of our body. And so that process of using oxygen isn't happening as effectively as we age. And this ultimately leads to a breakdown of cells and changes that we see in brains that are riddled with Alzheimer's disease. So the causes of these changes can be multiple, and we go into them in some detail in this interview. But the major contributor, in part, is simply getting older, right? So like we're at risk for everything when we get older, heart disease and cancer, et cetera. So it becomes more incumbent upon us as individuals trying to live healthy lifestyles as much as we can to kind of push back on these changes. Just like, if somebody's having joint pain or somebody's having heart pain or high blood pressure, doing the things that we always talk about, diet, exercise, sleep, stress, being aware of our environment, all help to push back. So one really, really important clarification I think that Dr. Gonzalez-Lima points out is that the earliest changes in the brain don't happen like they're explained in a lot of introductory textbooks and what is often a common understanding, that the changes in the brain happen in an area called the medial temporal lobes, which contain the hippocampus. Hippocampus is or the hippocampi, plural. That's an area that is thought to shrink, right, and often does shrink in classic Alzheimer's disease. But those are not the first changes. Those are much later changes. We first see changes in brain metabolism, right, brain energy. And those changes are picked up on certain kinds of imaging, right, that's not really accessible to the vast majority of people because it really is the domain of research, functional MRIs, fMRIs. And those changes are seen in an area in the middle kind of back part of the brain called the posterior cingulate cortex, or the posterior cingulate gyrus. And those changes seem to appear or show up quite a while before the atrophy. Not exactly sure, but probably years before you get real atrophy. And so there are two areas in the brain that make the hippocampus vulnerable, right? So the changes in the posterior cingulate cortex end up cutting off energy supply that ultimately leads to this sort of cascading of other areas in the brain breaking down. And we understand now the brain, as opposed to just being, hey, this area does this, another area does this, is sort of a committee or a network, a series of networks. And so when one area in the network breaks down, you get downstream effects. So when the posterior cingulate cortex, that middle area in the sort of back or top part of the head, the brain, starts to have trouble, it begins to cut off some of its support, called trophic factors, to other areas of the brain, particularly the hippocampus. Hippocampus also relies, it's a kind of an unstable area for blood flow, and then eventually you get that hippocampal atrophy that's so common or classic, because it's a classic feature in Alzheimer's disease. So that's how he explains or thinks about the vulnerability for the brain in Alzheimer's. And we talk a lot about this in the interview. And then, of course, we go through the key risk factors. What are the big risks for developing Alzheimer's? And he starts with and sides sort of with this camp of thinking that a lot of this is vascular. So it has to do with blood vessels, things that also cause heart damage. So all of the common things: smoking, poor diet, diabetes, high blood pressure, high cholesterol, sleep apnea and low oxygens, and then just a general decreased blood flow that happens over time as we age. And that decreases the blood supply to the nervous system, right? And then can lead to some of these changes that we see in Alzheimer's disease. So tests that may help kind of tell if you're having a problem early. So again, once real serious cognitive or memory symptoms set in, there's been changes for quite a while, right? So it's important to pay attention for those of us that are listening that are kind of proactive prevention people. Some tests that may help would be a carotid intima media thickness test that's sort of looking at the resiliency and squishiness of the some of the muscle structure around a blood vessel. And the more squishy and sort of flexible and pliable those are, the better sign it is for the health of the blood vessels. That's a good thing. And the other thing that is not done very often, but we do certainly where I work at Kemper Cognitive Wellness, which is EEGs or QEGs, brain maps, to pick up some of these vascular changes. It's not a perfect test, but we can really get an indication if are there vascular changes. We see a lot of these kinds of changes happening in 40s and 50s and 60s. So, maybe we are picking up some things that are happening in the brain, right? These brain network changes earlier before there's real serious, significant onset of kind of the full-blown dementia. Other risks would include, obviously, genetics, ApoE4 gene, which we've talked a lot about, head injuries and brain trauma. Certainly avoiding them is number one. And then if you do, somebody does have a concussion or some kind of head injury, to do something about it, and so you see someone that knows what to do about it. There are also some infections, viruses like HIV. Some of the herpes viruses are known to be associated with Alzheimer's, and just let us know if you'd like us to do some episodes about on viruses and infections in Alzheimer's disease. Please let us know. And then environmental toxins, like that's a new one that's now accepted worldwide by even the most sort of conservative Alzheimer's organizations and authorities. Environmental toxins, mitochondrial toxins, a lot of things in the air that we breathe, things that burn. Can gum up the works and sort of destroy our energy centers. So those are kind of the key risk factors. And then finally, the last couple of things we talk about, and this is really on the intervention side, and we've covered a little bit about this with previous guests on our show, is mitochondria, energy centers, and therapy. This therapy we're talking about is low-level laser therapy, also known as photobiomodulation. We remain fascinated by this because it's really a big sort of emerging area in Alzheimer's treatment. And this is where, to make it simple, and we talk about it in much more detail on the show, that light shoots photons. So the lasers shoot photons or energy packets, if you will, into the brain's photon acceptors, right? The laser light displaces electrons in an area called cytochrome oxidase. It's part of the machinery of the mitochondria, and it essentially makes more space, right? So these cytochrome oxidase, as a result of the lasers being fired, essentially change their state to one that's called oxidized or fully oxidized, and it makes them at that point more likely to bind and use oxygen. If you recall, the reason that's important is because from a brain energy perspective, it is Dr. Gonzalez-Lima's and others' theory that the main problem in the degeneration is the inability to be efficiently using oxygen. Right, so we talk about safety considerations regarding these lasers. Depending on the strength of the laser, the main risk really appears to be protecting your eyes. Dr. Gonzalez-Lima is using a 0.25 watt laser, which is really very low power compared to some of the other things on the market. And then the photons from the laser do seem to penetrate into the gray matter of the brain, but the white matter of the brain is more of a question. And then ultimately, Dr. Gonzalez-Lima, really, his hope is that we can do these not just with lasers, these kind of treatment, this photobiomodulation, but also with LEDs. And of course, there are other companies like V-Light that are already have trials underway, and you can get one of those yourself. The two lasers or photobiomodulation devices and companies that Dr. Gonzalez-Lima mentioned in this episode were Cyton, Cyton labs, and then CellGen lasers. And we'll have those websites in the show notes. And then finally, ketogenic diet. Of course, we always come back to the ketogenic diet, which is obviously a very high-fat diet. Working in even a somewhat damaged brain, somebody with cognitive impairment still has the capacity to receive, process, and run on ketones. We've seen a number of people improve fairly quickly, like in days to weeks, going from a kind of a usual diet to a ketogenic diet. It's a hard one to sustain. Don't really believe in sustaining it that long. I think most people have a cognitive benefit when they go keto. The question really is, how long will those benefits remain? And that's an open question at this point. But we mentioned coconut oil and MCT oil for energy conversion. And that really leads us into the next episode, which we'll talk about methylene blue. So I hope you get something out of this episode. It really is out on the edge of where Alzheimer's research is going. Enjoy. Welcome back to the show. We have another remarkable guest for you today. Professor Francisco Gonzalez-Lima is a world-leading neuroscientist doing research on the relationship between brain energy metabolism, learning and memory, and neurobehavioral disorders. He has contributed over 400 scientific publications in peer-reviewed journals, conference proceedings, chapters, and books. He's had his brain research funded for over 30 years and investigates currently the use of transcranial laser stimulation for cognitive augmentation at the University of Texas at Austin. Welcome to the show, Dr. Gonzalez-Lima.
Thank you.
Really excited to have you here. Clearly, you've had a distinguished and remarkable research career, particularly in the areas of brain mapping, brain energy, pharmacology, and several other areas. So you have, in my opinion, a uniquely informed perspective on brain and brain disorders. With that in mind, can you lay out what you think are some of the fundamental drivers or problems in the brain and nervous system and in the body in general that are leading to diseases like Alzheimer's disease?
Yes, I believe the important thing to start out is to differentiate between geriatric dementia or cognitive decline that is related with aging from what it is often referred to as early onset Alzheimer's disease. I believe these are two different disorders. They have different course, and of course, they have different risk factors. The major risk factor for geriatric dementia, that is referred to as Alzheimer's, is your age. And that, of course, is not the case for the early onset. And the truth is that this field of Alzheimer's disease is being uh, confounded by referring to dementia of multiple causations and at different ages as being a single disease,
Right.
Alzheimer's disease. So I don't think this is the case. Most likely, the early onset Alzheimer's disease, the early onset dementia that was described by Alzheimer's, is the original disease. And what we more commonly see, the majority, overwhelming majority of the cases, which are the geriatric or late onset dementia, are not the same disorder.
Right. So, in terms of the brain, if we talk about cognitive aging or sort of senile or geriatric dementia, what are you seeing as the fundamental problems? Where's the breakdown happening? Because obviously, both are a problem, but early onset, meaning Alzheimer's that typically runs deeply in family, often occurs before age 65, is distinguished from the so-called geriatric or um, Alzheimer's that we see more and more cases of as people get older.
Mhm.
From a sort of a brain energy perspective, or what is the problem in the sort of typical onset
Yes.
geriatric dementia?
Yes. For most cases, the geriatric dementia cases, one of the converging points that is related to the dementia is an impairment in bioenergetics. And what this means really is the ability of the brain to use oxygen to generate phosphorylation in a process that yields ATP, adenosine triphosphate.
That's
So,
sort of the energy cache of the we think about. They teach basic high school biology. It's the energy currency or energy cache
yes,
in our bodies.
that I think is a major converging pathway in the disease. What causes that then can be a lot of different things. However, for for sure, the major contributor is older age. In other words, there is a process that is happening as we grow older that allows vulnerability in the oxidative energy metabolism machinery that we have in brain tissues. And this vulnerability, if we have all the factors that play a role, we we may reach a point where we are no longer talking about decline of neurocognitive abilities, but a breakdown that will first manifest itself functionally in terms of energy metabolism, hypometabolism, but eventually will lead to atrophy and cell death.
Okay. So sometimes the way I think about it again simply is just like you see difference in skin or in joints in someone who's 25, 35, and someone who might be 75. Is it a similar sort of phenomenon in terms of brain energetics when it comes to sort of the Alzheimer's that's geriatric related, meaning that's primarily driven by age?
I believe that there are two processes going on. One is, like you indicated, is a more systemic change that is happening in aging in all tissues. And of course, the tissues that have higher demand for energy metabolism are more vulnerable when there's a compromise. And also, in the case of the brain, we don't have alternate sources of energy metabolism that do not rely on oxygen use. So the brain is more vulnerable than any other tissue to develop a problem, even though you may have the same systemic background happening throughout all of the tissues, the brain will show up. Uh, However, there is something else. Within the brain is not uniform in terms of metabolic machinery. Different neuronal populations have different vulnerabilities. They use energy in different ways. Neurons are large, have large cell bodies and large axons. They do not have the same kind of vulnerability that smaller type of neurons, like we call interneurons, for example.
Hmm.
When neurons serve other different purposes, they have to then share some of that energy for different functions. So we have a heterogeneous or mosaic type of vulnerability that manifests itself in different ways. And there is a misunderstanding in the literature, especially introductory textbooks, about where in the brain you see the initial changes in Alzheimer's disease in these geriatric populations. And I like to clarify that the initial functional changes, the ones that are related to hypometabolism, are not in the temporal lobe, in the hippocampal area. They are in the posterior cingulate cortex. So this is a region that shows the largest and earliest onset of hypometabolism in individuals who later on develop geriatric dementia of the Alzheimer's type, or individuals who are transitioning through so-called mild cognitive impairment. And this is something that is important because there are groups that are focusing on trying to prevent neuronal death and talking about areas where you can see atrophy, anatomical atrophy. It is too late at that point to try to rescue those regions of the brain. You have to intervene before the hypometabolism, and therefore you have to make sure that those areas that are going to be the first one revealing that change can be rescued.
So that's interesting. Let me follow up with a couple of these questions because you kind of are leading me into where I was hoping this conversation would go. So if I understood you correctly, first of all, obviously not everybody who's 75 years old needs will develop Alzheimer's, but somebody who's 75 years old is at higher risk of Alzheimer's, just like somebody who's 75 years old is higher risk for heart disease and cancer and needing a joint replacement. So part of this is, why do some people, or why are some people vulnerable? And if you have an answer to that question, that's great. If not, I think that's probably still an open question. But why are some people's brains and neurons more vulnerable? But
Mhm.
the other, do you have an answer to that question?
Oh, yes, definitely. We know for sure that there are a number of risk factors that will add up in a cumulative way to compromise oxidative energy metabolism. And the most clear one that is related to aging is as we grow older, we have a progressive and consistent decrease in the blood supply to the nervous system, especially the brain. And this is happening, is measurable. You can see at least from decade to decade, you can see significant differences in this decline. So there is a vascular risk factor that not only results in dementia, it also, as you know, it can lead to all kinds of problems
Hmm.
in older age. But I would put like a number one. And the reason I say that's number one because that's happening essentially in everyone who is aging. It's happening in different degrees in different people, and so the progression doesn't have the same rate. But it is a constant for anybody regardless of individual differences. But then beyond that, then we have our constitutional makeup. We have different genetic backgrounds that result in a different structural constitution and also result in our functional machineries operating differently. And as you know, uh, genetic risk factors are also found that can make people more vulnerable
Yeah,
for late onset Alzheimer's disease, like having these ApoE4 alleles, one or two,
sure.
especially having two, increases your chances. But that doesn't mean that that causes Alzheimer's disease. It just adds up. That is a genetic risk factor for all cardiovascular problems that have been looked into. So this just adds up. to the issue of cardiovascular compromise. And in addition to those, there are more incidental changes that are not related to your family background or your age, such as having trauma to the head that may lead to brain injury. All of these traumatic events will add up to increase your risk for dementia. And then you can have other insults. And we all know that we can be constantly subjected to infectious agents, and they're going to have an impact, cumulative impact over time that will compromise the brain. We know for sure if we have an infection that reaches the brain, let's say a viral infection is produced by HIV,
Hmm.
we're going to end up with HIV related
Yeah,
dementia.
yeah, frontal temporal
So
type. Yeah,
there is no question that each of these factors by itself is sufficient to produce dementia. So, in other words, if you have purely vascular factors, you end up with vascular dementia. If you purely have trauma, you can also have, a post-encephalitic traumatic,
dementia.
yeah, type of dementia, and or what used to be called dementia
Yeah,
pugilistica, what the boxers
sure.
and other people getting hit in their heads will develop, and the same thing applied, like I said, also to infectious agents. And we don't know for sure, but it appears that if you live long enough and do not succumb to any of these other factors, you may end up with senility,
Hmm.
uh, and that will lead to senile dementia, even uh, if you didn't have any of these other uh, vulnerabilities. So at some point, any of those can contribute, and all of them, especially, can make a good case for what's happening.
Yeah,
And then the only other sort of major type of insult that I will bring up is related to environmental toxins,
yeah.
in particular, environmental toxins that are linked to oxidative energy metabolism. In other words, mitochondrial toxins.
What are the big ones?
Toxins. Mhm.
What are the big ones of those that come to mind for you?
Okay. Since in mitochondria, the electron transport is the key in terms of the interaction with oxygen. So toxins that interfere with some of these complexes in the electron transport can by themselves lead to dementia. For example, uh, in something called Leber's disease, that you can have a toxic compromise of cytochrome oxidase complex 4 of the mitochondrial electron transport, and that is sufficient to give you a case of dementia. You can have the same kind of disorder with the same syndrome if the compromise to complex 4 is due not to an environmental mitochondrial toxin, but due to a genetic mitochondrial defect.
Right.
So, unfortunately, uh, it can be more than just cytochrome oxidase complex 4 that is compromised. There could be anywhere uh, of complex 1, 2, 3, 4, or 5. However, it appears that whenever we look into brains, and I have done this of individuals that have died and had geriatric type of Alzheimer's disease confirmed pathologically, that the major biochemical change that I was able to detect was an inhibition of this complex 4. Even though in different individuals you will have other problems, this was a common denominator. and the region of the brain where the largest inhibition uh, occurred was that posterior cingulate cortex.
So let me slow down because we got deep down the rabbit hole, and I'm super interested. I just want to make sure all of our listeners are sort of tracking with us. So I'm going to try to forgive me, but I'm going to try to oversimplify this. One of the things that you've uh, made a major contribution to discovering is that Alzheimer's, at least some kinds of Alzheimer's, and it's important to recognize that there's probably a lot of different personalities or subtypes or phenotypes of Alzheimer's, and we've discussed that in previous episodes on the show. But at least for some kinds of Alzheimer's, there seems to be a problem with blood supply, cerebral blood flow, and then there's this impact on the brain energy, right? And we've talked about, the
Yes.
mechanism by which we make energy, ATP as it were, is through the mitochondria, and they have this series of complexes. So when you're referencing complexes 1, 2, 4, etc., so you're representing, you're referencing rather, sort of various stops along the trail of energy production, correct?
Yes, and more specifically, so I will say we are referring to the movement of electrons along these complexes, and that trail, that movement of electron, is destined eventually to reach oxygen and reduce oxygen into water. In other words, oxygen acts as our ultimate electron acceptor, and what we have found is that in mitochondria, this process of accepting electrons, the transport and the accepting of the electron by oxygen, is what it leads to oxygen consumption. The overwhelming majority of the oxygen that we breathe is used in that reaction, which is catalyzed by that cytochrome oxidase or complex 4. The key, what makes that important is that that is coupled to the production of the ATP, the so-called oxidative phosphorylation. So you can have so that route that you're describing is the route of getting those electrons into oxygen. And where does the electron come from? The regular electron donors that we have come from what we eat. So the foods that we eat eventually are transformed into electron donors to feed this electron transport to reach oxygen that is coupled with oxidative
Very
phosphorylation.
interesting. I actually want to come back and talk about. We'll talk about, as we transition in this interview to talk about potential therapies for Alzheimer's. We'll talk a lot more about electrons and oxygen being sort of the ultimate electron acceptor. But I did have a couple of other questions on the assessment of blood flow. You mentioned that,
Mhm.
classically, and this data has been around, as you know, for a while now. Um, One of the classic biomarkers, as it were, for, hey, this is the picture of Alzheimer's, is that there's atrophy, there's loss of brain tissue, particularly at the hippocampus, but it could be other places as well. It's a deeper part of the brain. But you
Mhm.
mentioned a posterior cingulate cortex, right? Um, and you mentioned that that's
Yes.
sometimes an earlier point. Where, and how does that show up? What
Uh,
kinds of studies?
yes, that's a good question. Let me clarify. Not sometimes, every time. The hypometabolism in the posterior cingulate cortex always, in every case that have been looked into, precedes the atrophy that takes place in other parts of the brain. But the brain is a network of interconnected regions, and essentially, one way that you can link the two findings that you just mentioned is if you have a functional decrement, a hypometabolism in the cingulate cortex, especially that posterior cingulate cortex, all the way to the so-called retrosplenial limbic cortex. That region provides the major input into the so-called entorhinal cortex or parahippocampal cortex. And that parahippocampal cortex, in turn, provides the main input into the hippocampal formation. So what you do when you functionally deactivate the uh,
Yeah.
cingulate, especially the posterior half of the cingulate gyrus, you are is functionally denervating the parahippocampal gyrus, which is the major input to the hippocampal formation, and then those regions are going to atrophy. Uh, but they're not atrophying because something is happening intrinsically in them, but is because they're losing the trophic action that is what is exchange when a neuron synapses with another. It provides a trophic action that maintains that other neuron alive. It maintains the excitability, allows the exchange of ions. And so what we are missing here is in the simplified picture of Alzheimer's, we're looking at the
Yes.
consequence of an event that is happening earlier on. If you denervate the muscle from from the axons that are in the neuromuscular junction, that muscle will atrophy. The same thing happens in the brain. If you, of course, in the brain, you have regions that get inputs from many other regions. But if you have a region like that parahippocampal cortex or the hippocampal formation that essentially have a very clear dominant major input from that pathway, then if that pathway is compromised, is hypometabolic, is no longer sending out those outputs, those regions are going to atrophy.
It totally makes sense. So basically, you're cutting off a supply in one region of the brain. We're calling it the posterior cingulate cortex, and then then you're sort
Yeah.
of cutting off supply to an area of the brain that eventually
Mhm.
shreds, and that's very much related to the kind of classic short-term memory problems.
And unfortunately, well, 2 points. That network property of how many inputs a region gets defines vulnerability as well at that level. A region that gets input from more regions than another has less chances of being compromised if one of those or more of those inputs is compromised. So that's what makes vulnerability at the network level. And then, unfortunately, things are not always completely straightforward. Uh, we do know that some areas of the brain are more vulnerable to loss of blood supply or loss of oxygen delivered in the blood supply. Even if we subject the same regions, we can do experimentally to the same levels of so called ischemia or loss of blood supply
Hmm.
or perfusion to an area. Some of them are more vulnerable. Those neurons are going to resist less time without being able to use oxygen. One of the reasons is we have very limited energy reserves in neurons. It's not like other tissues, for example, that you can have glycogen to accumulate in any significant
Yeah.
levels, like in the case of muscle, we have large amounts, or in the liver. In the case of neurons, it's a minimal amount of glycogen that you have, and some neuronal types have less than others. And guess what? Any pathologist will know during training when I used to teach medical students anatomy and neuroanatomy, and also the residents in pathology, they know that if you diminish blood supply to the brain, or even if it is overall, the most vulnerable region to ischemia and hypoxia, that is, decreasing blood supply or decreasing oxygen in the blood, it is the region of the hippocampus, the part of the hippocampus that is called the hippocampus proper or
Yeah.
the CA sectors. So whenever you have anybody that has compromised, that's the first area that's going to atrophy in the brain from a pathologist point of view. So we have this vulnerability on top of the other mechanism that I was telling you. So that hippocampal formation is now affected in two different ways. It's affected by the lack of inputs from its major source that provides trophic excitability, and also is less likely to sustain hypoperfusion to the
Yes.
brain.
So that's an important clarification. So then, is there a is there a practical way to assess for cerebral brain flow, and particularly looking at what might be happening there in the PCC?
Yes, I would say the most obvious way to deal with this geriatric dementia is to worry and evaluate and intervene in terms of cardiovascular health. You want to maximize the ability of the cardiovascular system to provide oxygenated blood to the brain. And this can be done in any number of ways by normally as when we're growing older and we're losing elasticity in every elastic fibers that we have in any tissue in the body, including the ones that are wrapping around our arterial blood vessels.
So like a carotid intermedia thickness exam, something like that.
Yes, yes. By looking at the carotid thickness, especially those layers, you can predict the level of cognitive decline, even in healthy middle-aged people. I have participated in this kind of work in the past with a colleague, and it is possible already to see that relationship. So, what can be done in practice is try to maintain that blood supply, try to maintain that, trying to maintain those blood vessels from developing these thickened walls, from decreasing their lumen, the hole where the blood is going through, uh, and to facilitate their elasticity, how's the heart pumps so that they resist less to the pumping, which the more they resist, the more arterial pressure goes up. So if you can control, relax some of these sources of resistance, all of that helps because normally we have an auto-regulatory mechanism that is dealing with this as we are growing older. Starting as an adult, we get this process going, but at some point that auto-regulation is not enough.
Yeah.
That's the point. The point where you can see arterial blood pressure, hypertension has a disease. So dealing with that, dealing with what generally referred to as the metabolic syndrome.
Yeah.
If you have a problem with being able to use glucose by tissues, especially by the brain, the major consumer of glucose, and what is that? Why is that glucose important? That glucose is going to be used to generate those electron donors for that mitochondrial electron transport that is linked to the use of oxygen for energy production. So
Hmm.
if anything interferes with that substrate, the glucose or any other substrate that eventually leads to the formation of uh, electron donors, you need to try to fix that. So, treatment for diabetes, insulin, and extremely important for a medical condition that we call obesity,
Obesity.
because
Yeah.
obesity provides, unfortunately, all of these changes that are detrimental for cognitive function. So, obesity, even if you're dealing with young adults, uh, we done it there, it will interfere with cognitive performance. And of course, it's has to do with this metabolic syndrome. So I believe that there is a lot that can be done in practical terms by taking care of our cardiovascular function, taking care of endocrine function, so that you can use your substrate for the initial steps in the energy metabolism, and maintaining body weight to prevent hypertension and prevent this diabetic syndromes. So a lot can be done there.
Can I ask you a it's just a follow-up question? This may be a little out of your wheelhouse, but I don't. I think from what I read, it may be within. There's a number of people. You've had numerous studies using functional MRIs, which we don't necessarily need to get into, but that's not obviously a practical, accessible tool for most people at this point. It's more in
Yes.
research circumstances. But one relatively practical tool on the assessment side of different regions of the brain, although it doesn't have it's not like doesn't have the resolution, so to speak,
Hmm.
of MRI or fMRI, is EEG, right? So looking at
Hmm.
alpha frequencies, and there are a few papers. I think who was it, Wolfgang Klemesch, and are publishing on peak alpha frequency, and
Hmm.
without getting too technical, but there is there are some suggestions when the waveforms uh, in alpha, so to speak, on an EEG, like we do EEGs in our office,
Yes.
when those start to change, it is an indication of some kind of potential vascular insufficiency. Do you have any thoughts about that?
Yes. I agree. It is possible to pick up some of these changes in various waves. To focus on the EEG, the electroencephalography, just picking up the electrical voltage changes from the scalp. The problem there is you have to do a little more sophisticated signal processing to be able to detect these changes. But nowadays, this is
Yeah,
all within reach of practitioners, and you're absolutely right. The effects are again a reflection not of an individual just region, but they are the result of a network operational change, and that since our levels of arousal or levels of consciousness is pretty much reflected in terms of the frequencies of the EEG, the so-called brain waves. How often do they oscillate up and down per seconds, the cycles per second. This particular alpha state that is around 10 times per second in terms of oscillation, or 10 hertz, plus or minus 2 or 3 hertz, seems to be the kind of reflection of a brain that is awake and is attentive, and it can perform cognitive operations more efficiently than if we have a brain that is oscillating in terms of these electrical waves in any other, especially less frequent than,
10
than
hertz.
10
Yeah.
times per second. And so it is a way to monitor whether what you're doing is improving somebody being able to show this alpha frequency in the resting state. And the alpha waves are present everywhere in the brain. Usually they talk about
Yeah,
the
posterior. Yeah.
occipital cortex, but in reality, we can monitor them, for example, in the prefrontal cortex, and those in particular will be more related than the occipital ones with this cognitive processing, especially prefrontal based type of cognitive
Yeah,
processes.
so that's interesting, and I wasn't planning on spending time, but since you mentioned it, so right, as you,
And we can modify that since you're interested in talking about other interventions. Uh, this intervention that we're doing using laser
yeah,
light, yeah, in the so-called near infrared or lower part of the infrared spectrum, like what is near what is called 1000 nanometers wavelength. Well, that kind of low level in terms of the intensity uh, of the laser light, so it's innocuous to tissues, but it is sufficient to increase alpha waves in the prefrontal cortex. And we measure this directly, and we were the first to describe this phenomenon. This use of near infrared light is generally referred to as nowadays as photobiomodulation. Before, it used to be called low level laser therapy or low level light therapy when done with uh, so-called LEDs, light emitting
Hmm.
diodes. And so we can directly assess, for example, by having somebody undergoing EEG, if an intervention such as stimulating their forehead with laser light in the near infrared range can increase their so-called alpha power, that is the density, the amount of alpha waves as relative to the other waves that are present in the EEG. and and it takes only a matter of minutes to get significant increases over baseline. And now we have done this m- many times. We have only published a couple of studies, but we now have done this many times in our labs. And the other thing that we can use to monitor the effect of something like the photobiomodulation is to actually measure the amount of oxygenation that your blood is carrying, especially to your the region below your forehead, the prefrontal
Yeah.
cortex. Remember that I talked to you about the posterior cingulate,
Yes.
and now I'm talking to you about the prefrontal cortex. Well, the prefrontal cortex is the second one in line to show these hypometabolic changes in the Alzheimer's cases. So you have number one posterior cingulate, then number two prefrontal cortex, and then number three you have posterior parietal, and then only number four is the medial temporal lobe regions in terms of functional changes.
Yes. So that's
So,
exactly, you just answered my question. So that's extremely important. So posterior cingulate, prefrontal cortex, posterior parietal, and then medial temporal. So
mhm,
the reason I feel like that's important is because I think we, for all the reasons you said, we can look at kind of lab blood test risk factors, family histories, certain amount of genetics seem to come into play, and then for those of us that are kind of really trying to take a deeper dive, and someone, let's say, is not neurologically abnormal enough to justify an insurance company paying for an MRI, sometimes we'll get an
mhm,
EEG, and you can get these sort of early readings on people, and you can definitely look at things in general. Again, the spatial resolution is not wonderful with the EEG, as you know, at least not a 19 channel
sure,
EEG, but meaning it's not a clear picture, and but you can get a
mhm,
general sense whether something's happening more towards the front, like you mentioned, or more towards the back, or
mhm.
perhaps in this sort of temporal medial area, sort of the areas underneath your
Yeah.
jawbones and towards your nose, things like that. That we talk.
I I agree completely with you. I think there is a really important role for integrated medicine in dealing with dementia and what is referred to as geriatric or late onset Alzheimer's. I think the approach that you're presenting is the way to go. You need to have an evaluation that involves blood biomarkers that tell you about the health of the cardiovascular system, that they tell you about energy metabolism, and you need to get also some neural functional indices, starting out, for example, like what you indicated, looking at alpha. However, I have to say, ideally, like what we see, the major changes in alpha power, but also we see an increase in beta power, a much less increase. This is important because if you're just changing the level of consciousness of someone by the intervention, you're going to see more or less alpha. However, you're not going to see more alpha plus beta if you're just simply having somebody who is becoming more relaxed and is becoming more synchronized from during the intervention.
Let me clarify. I think I didn't follow you. You're saying, in addition to having, let's say, abnormal alpha brainwaves, people also have abnormal beta brainwaves in cognitive impairment?
No, what I was saying was when we administer
Okay, when you're using the
photobiomodulation,
laser photomodulation. Okay.
yes, we can improve primarily the alpha power, increase the alpha power, but secondarily, to a much lesser degree, one also improves the beta power, which means we're not simply making the people more relaxed, and
Yes.
we are indeed putting the brain in a
Of
state
alertness.
that favors cognitive
Yeah,
processing.
alertness, getting stuff done. Do you know in what, and again, I apologize to our listeners if we're getting overly technical, but it is important. Do you know in what frequency band range? Are we talking like 13 to 15 hertz? Or are we talking about higher beta, like much faster than 15 hertz?
Yes, it's usually no. It's usually
Low
the
beta.
lower
Okay.
half of the beta
So toward a 12 to 15
range.
range, sort of, so to speak. Okay. So
Correct.
that's,
Right. It's really like a continuum. As you move increased frequency over the 10 hertz, and then you get into the beta
so
range.
when you're using the laser, Francisco, and are you working with people that are how cognitively impaired are some of these people that you're seeing
Well,
improvements in or seeing the changes, at least physiologically?
yes. First of all, we started out with healthy young
Sure.
individuals and to
Yeah.
validate two things. One, that we could improve a number of cognitive tasks since we are applying this to the forehead. And the reason we're doing that is because there is no hair in the forehead that will absorb the light. So through the forehead, we can get more of this light to penetrate to the cerebral cortex. At least it penetrates as deep as the gray matter, the surface of the cerebral cortex. And in that case, if you do prefrontal based type of cognitive functions like vigilance, sustained attention functions, or so called working memory
Hmm.
functions, and you can detect the effects of the laser right away if you look at before and after stimulation, or if you have groups that are given placebo and stimulation as compared to the active one. Then we look into adults and
Can I clarify
and
one thing you said there, Francisco?
yes.
Did you say you're doing the laser and you're giving them some type of cognitive task?
Yes, we the first thing that we did with the laser was to verify that indeed at the behavioral cognitive level, uh, our dosing that came from our animal studies uh, was effective in making a difference in terms of cognitive performance. And
Hmm.
then the second thing we had to do was since our molecular target for this photonic stimulation, these photons are provided by the light, are absorbed by molecules inside the body. So, where are these molecules? Which is the one? So, inside cells, especially neurons, we have a lot of mitochondria. And these mitochondria contain the most important photon acceptors inside cells. In particular, the number one photon acceptor is that complex 4 or cytochrome oxidase. In fact, that's where the name comes. Cytochrome, cyto means cell, chrome means color. So by color, we mean when a molecule absorbs particular wavelengths of light and then reflects the other wavelengths. That reflected wavelengths, the one that gives the color in the case of visible light. So cytochrome oxidase is the if we provide the same amount of photons to tissue, the main major intracellular sector is cytochrome oxidase, and it becomes oxidized. These photons, these what they do is they displace the electrons that are present in cytochrome oxidase. Cytochrome oxidase all the time cycling electrons is part of the redox cycle. Well, what it makes it so cytochrome oxidase lives in multiple stages between fully oxidized and fully reduced. The more fully oxidized it is, the more affinity it has for oxygen, and vice versa when it's fully reduced. So what the light is doing at the molecular level is targeting primarily cytochrome oxidase inside mitochondria and making it increasing the relative concentration of the oxidized fraction of the enzyme, which makes the enzyme uh, increase its affinity to interact with oxygen. And when it interacts with oxygen, because that process is the one coupled with oxidative phosphorylation, then we supply more energy. We increase uh, oxygen consumption by the brain. And fortunately, that process of use of oxygen, when the oxygen starts being used, the oxygen supplied by the blood, there is a relationship that results in a so-called hemodynamic response. What that means is that the same cytochrome oxidase releases a gas called nitric oxide that vasodilates the local capillaries where that oxygen is being extracted from the blood and allows then more fresh blood with oxygenated hemoglobin to resupply the region. So this is the so-called uh, vascular coupling that we have between the energy utilization and the supply of oxygenated blood. So that process is also facilitated, and we can measure that. For example, this is what took us sometimes. I have to team up with bioengineers to the methodology to be able
Yeah.
to directly monitor non-invasively through the forehead uh, what is the impact that we're having on cytochrome oxidase. And indeed, we published this in the Journal of Cerebral Blood Flow and Metabolism that you indeed increase the concentration of oxidized cytochrome oxidase. You increase, then after that happens, then you increase the amount of oxygenated hemoglobin that is delivered locally. So you can demonstrate the physiological responses to that photonic stimulation. I believe in the future, and this to do it in a practical way with the technique we use is called broadband near infrared spectroscopy may not be very
Yeah.
feasible. We have a custom made system. However, there is a simpler technique that is called functional near infrared spectroscopy that is able to detect the
Yeah.
oxygenated hemoglobin and the deoxygenated hemoglobin relative ratios. So that one can be used. FNIRS is the abbreviation. FNIRS have the potential to be used as a way to detect whether your intervention is increasing the oxygenation to that prefrontal
And
cortex.
that's I assume fairly real time. I mean, you can. Are you?
It is in real time. Sampling rates are usually about 10 hertz or more.
Okay.
10 sample 10 times per second. We don't need that kind of temporal resolution for what I'm telling you because this is a process that takes actually minutes.
Yeah.
So
So I don't have
you
to be
can
that fast.
integrate a few seconds at a time, and however you can see. So we have looked into this in older people that have a memory complaint. In other words, these people don't have yet diagnosis of mild cognitive impairment, even though some of them did reach the our criteria when after they were recruited. But in that population, we found a beneficial effect of in terms of cognitive performance, and and now we have a paper that is under review where we are showing what is happening in terms of this broadband near infrared spectroscopy measures of cytochrome oxidase and oxygenate the hemoglobin. And one thing that I can anticipate to you is that uh, they do have a response in their mitochondrial response with cytochrome oxidase increasing its fraction of oxidized uh, cytochrome oxidase, which is a good thing because that's one of the things that is happening in old age is that we're losing these mitochondrial enzymes in terms of activity levels. And however, the hemodynamic response, the vascular response, is not as large, as effective as what happens in the younger uh, samples.
Okay, so
So,
let me clarify. I just want to, because, and then I want to ask you a question, but I want to clarify the if we just to try to again to summarize it just a bit. So you're essentially using these low intensity lasers, the front part of the brain, to impact the mitochondria. You explained cytochrome and how you're captured this, how the mitochondria elements of the mitochondria sort of suck up or accept the photons, the energy that the laser is
correct.
providing, and that's having a positive impact on oxygen or oxygenation to so as to, among other things, improve blood flow. Is that an accurate?
Yes, besides improving oxygen consumption coupled to ATP production, you also have coupled to that process. As soon as you start losing the oxygen locally, then you have these vasodilation mechanisms that are coupled with it to replenish oxygenated blood to the area. And in fact, that's a response that is about 10 times larger than the amount of oxidized cytochrome oxidase. In other words, we always overshoot the hemodynamic or vascular perfusion response is many times larger than the actual energy that has been consumed uh, by the tissue. And this is why nearly all of these brain imaging techniques use hemodynamic signals uh, because they are larger than any other signal that you can get from the brain.
Okay, like fMRI, for instance.
Yes,
Yeah,
all of the fMRI, the so-called BOLD
bold. Yeah,
signals, blood oxygen level signal, that's what it stands for.
yeah.
They use blood oxygen level dependent signal. That's where the ball came from.
Yes.
And however, with the F nears or the BB nears that we use these techniques, one we don't have the ball is a compound signal. You can't tell about how much oxygenated versus deoxygenated versus just blood flow is present in the signal. It is a compound signal because it maximizes the size of that signal, and those signals were very small. That was the only way you could get anything out. However, with the F nears, you can separate out the oxygenated hemoglobin, the oxygenated blood from the deoxygenated blood. So you can see essentially the oxygenated hemoglobin going up after the laser stimulation and the deoxygenated remaining low or going down, depending on how quickly you have blood flow going to the area. Because let me remind you something, having blood flow to an area is not enough.
Yeah.
If you just stop breathing, you hold your breath, you're going to increase blood supply to your head, to the brain primarily. But that blood is not going to be oxygenated. So
Yeah.
that's the important thing about, for example, the photobiomodulation with this laser therapy is that you are increasing the amount, not just of blood flow, but of oxygenated blood to the brain tissue.
Right. That's the key. Right. The key, of course, is the oxygenated brother. Because that's where the magic happens. Are you?
Yes,
Are you?
because some people say, well, we have pharmacological agents that can increase blood flow, but that helps only partially, because if you cannot increase the net amount of oxygenated,
Right.
it's not going to make a difference.
Totally. So you got to treat all the reasons, right? So the meds help, but you also have to deal with the oxygen problem. That's interesting.
Correct.
So a follow up on that. Are you familiar? I mean, I'm sure you're familiar with Russ Swerdlow's work and he's really looking at how do you assess um, mitochondrial function. I don't know. He's part of the so-called mitochondrial mafia. We've had other members of the
No,
mafia on the.
I'm not familiar with any mafia
No,
members.
he's no, he's the chair of the essentially Alzheimer's research at the University of Kansas. A real smart guy.
Oh, yes, yes,
Yeah,
yes.
Russell
I am
Swerdlow. Yeah.
familiar with his work, and I have visited him and gave presentations there.
Yeah, I have too.
Uh,
Real smart guy. I thought the
yes,
this
yes.
pairing would be work.
I am so happy that they're doing the work they're doing.
And the reason I mention his work is because I initially learned of him as somebody, he was trying to figure out mechanistically what a ketogenic or a very high fat diet does, and its impact on mitochondrial function. And so, are there
Mhm.
roles for things like in extra oxygen? Obviously, I'm really aggressive about looking for sleep apnea and oxygen problems because we see that that obviously people's brain performance picks up. Even people that already have somewhat broken brains can have sometimes minor, sometimes much more significant improvements in mood and then
Yeah.
attention and sometimes even memory when you
Mhm.
treat that. But do you see things like obviously CPAP and oxygen, but even things like a ketogenic diet and or
Yes,
hyperbaric oxygen?
you are absolutely right. First of all, you do whatever you can to improve oxygen metabolism, because that's the primary one. However, uh, one of the problems that we have as we grow old, and this was discovered in rats, older rats, and then verifying humans, is for example, when we're talking about the glucose, as we grow older, the glucose transport into neurons is diminished. It doesn't matter how much glucose you have circulating, it doesn't get transported into neurons. So then it's a chain of events. If that doesn't happen, then you don't get electron donors, then you cannot provide those electrons to oxygen, and that process is not coupled with the energy production. So yes, I agree. Find ways to improve that. Of course, one way to improve, let me just expand very briefly on that. If then you have more glucose that is circulating that cannot get to your brain or other excitable tissues like muscle, then you're going to have high blood sugar, and you're going to have increase in insulin release to try to deal with that, so that you can facilitate the transport of that insulin into tissues. But if you chronically have this situation, these insulin receptors
Stop responding. Not as responsive.
start responding. So then you develop the insulin resistant type of diabetes that these older people or metabolic syndrome people show. So all of it is related. So your integrated medicine approach is uniquely suited to look at these various aspects. So yes, first try to improve that phenomenon. However, to tie it up with the ketogenic diet, one of the things that is found in rats, in older rats, just like older people, even though glucose transport is impaired as we age, the transport and production, well, in this case, like the liver producing these ketone
Yeah.
bodies, uh, it is not impaired. And the capacity of neurons to use those ketone bodies to derive energy uh, in using still the mitochondrial machinery, but not in the same way, is still present. So therefore, you could have an alternative way to supply energy to the brain. We normally, under physiological conditions, we always engage in some degree of ketogenesis. When during the fasting phase of our day, when we go to sleep
Yeah.
uh, and we spend 8 hours without eating, we are engaging in this ketogenesis. And then our brain is relying on some of that, especially early mornings, when glucose levels are small. And
Yeah.
if you are older, then you are even less efficient in using them up. So you're relying on that. And developmentally, babies, when they're born and they are breastfed, during the entire period of lactation, breastfeeding, they're primarily using ketone bodies from lipids, especially these medium chain triglycerides,
Hmm.
has the source of energy for the brain during physiological development uh, postnatally. So this is happening not only in development, but it happens every day when we go into a fasting stage. So I agree with you and my colleagues in Kansas City that it is a good idea to try to improve ketogenesis and to provide that alternative source of supply. And I, as you probably know, things that are so simple as coconut oil provide a large amount of this medium chain triglyceride. Most of this of the oil in the coconut oil will contain that type, and that type of fat uh, is primarily used for energy conversion, not for storage or structural type of applications. It is primarily used for energy conversion. So I definitely believe that's one alternative that should be part of an overall treatment plan.
Absolutely. I want to just circle back for one or two more points on the lasers, if we could. Photobiomodulation we were talking about before. Is there any danger to the lasers? Most people think about, wow, shooting a laser into my brain. That
Mhm.
that seems dangerous.
Mhm.
What are the safety considerations when you start up with a laser?
Yes.
Because I know there's some things that are sold over the counter. I mean, basically they've got FDA exemptions because they're so low power that they're
Yes.
not considered dangerous.
Yeah, that's a very good point. Yes, most of these low-level laser devices are so-called FDA clear, that they pose no risk to humans. However, and we're surrounded by lasers nowadays.
Right.
Everybody seems to have laser pointers, and even children are now, playing with lasers
Yep.
and dancing and getting lasers shoot at them. So the only risk is risk to have the laser pointed towards your eyes, because the optics of the eye allow the convergency of all of those rays, that of wavelengths of light into one point in the retina,
Yes.
and that can increase then the effect and produce damage to the eye. So when we do this, people have to wear dark glasses. They look like sunglasses, but they selectively block the wavelengths, a range of wavelengths that contains the ones that we're using. So that's really the only laser safety factor. We operate this at what is called power density. That is the amount of milliwatts per centimeter square per surface area, that doesn't do anything to tissues. So we're below those thresholds. We're usually like one quarter, 25% of that threshold. When
What is the wattage you're using? What's the power you're using?
it's a 0.25 watts per centimeter square.
So pretty low power. That's amazing. Because I know there's some controversy about whether that actually travels, through the skull and
Yeah, it doesn't make any difference. It There is a little bit of a misunderstanding that if you have more power, it's going to travel longer distance, and this is not true. Uh, the distance that it will travel is a function of the wavelength and how that wavelength interacts with the tissue, and the major variable there is when the light gets dispersed, has it enters the tissue, and that will happen with one photon or 1,000 photons to the same level of dispersion. So obviously, the amount or intensity that will reach the deeper parts will be smaller if the initial incident light
Smaller.
is smaller,
Yeah.
but it's not going to affect the penetration. You're just going to have less of it reaching the same depth. And now we can measure this directly in models of the brain, or models of brain tissues, where we put together things that absorb light in the same way as different parts of the head.
Yeah.
And we can also measure this in animals more invasively, putting probes at different depths. And so we are very certain. I have also looked into cadavers, but they have a lot of limitations. But where we have passed this light and measured across the skull and so on,
Yes.
we are pretty certain that it penetrates, and it penetrates through the gray matter of the cerebral cortex.
That's impressive.
The white matter is another thing. The white matter presents more of a barrier to these photons. So it more or less defines then that it is the gray matter of the cerebral cortex when what becomes the regional tissue target for this transcranial laser stimulation. We still don't know whether using the LEDs that are not laser, then the main difference is that the lasers are essentially of only one frequency. When I was saying like 1,000 nanometers, the wavelength is
Yeah.
the same. And the LEDs have uh, a small range. Depending on the quality of the LED, they have a smaller or larger range of wavelengths uh, that are taking place. So there is a little bit of controversy. It appears that in most applications, both can benefit from it. There are other people, there are things that do not happen with light uh, that is not uh, in the way of laser light. The laser light doesn't open up, it's collimated,
Yeah.
and the there's a phenomenon that happens with that laser that doesn't happen with others. So it's still I think those will be empirical questions that one have to resolve. My hope is that we will be able to do this with LEDs that will simulate the same kinds of parameters. Unfortunately, there are a lot of inexpensive LEDs out there that do not do that, or that they quickly become very inefficient. The way you can tell, the electrical power that you give into an LED needs to be transformed into optical power, the amount of light that comes out.
Yeah.
But the more inefficient that device is, the more heat it generates. The actual LED heats up, the light source heats up. So that means that more of that power is dissipating as heat instead of being converted into optical power. And the in devices are not high quality like the ones that are found uh, inexpensively on the web.
Yes.
Uh, they may start out with that nominal amount of optical power. However, very soon after seconds, they start heating up and dissipating heat, and they are no longer in the therapeutic range in terms
Mm.
of the optical output. And I have actually measured some of those devices, especially the ones that were initially used for intranasal stimulation, and they all had that same problem, and you ended up with very small amount of optical output. So we measure these all the time, and we measure not only what is nominally supposed to do, but we empirically see that that's the optical output, because every device will have a different efficiency level and will dissipate heat in more or less degree.
Are there any over-the-counter devices or direct-to-consumer devices that you feel like are sort of on the level that are actually most likely doing what they're saying they're doing?
There is a company that I have to disclose. One of the former trainees here from the University of Texas at Austin, uh, in the he got this PhD in engineering, and he developed lasers for my lab, and he developed other devices for my lab. And now he took some of that expertise and came up with a company. I think it's called Cytosys, or Cyton, like Cyto from cytochrome.
Yeah.
uh, and so the reason I liked that one, and I serve on his scientific advisory board. I get no funding or any thing from doing that, but he's a former trainee. Was one of these doctoral dissertation committee members in the Department of uh, Electrical and Computer Engineering.
What's that
So
called? What's that company called? Cytosys?
it's called Cyton Sys, CITONSIS,
Okay.
Cyton Sys, and that's the one that this company startup is here in Austin. There is another company in Dallas that we have been using their lasers as well, and I, because we use those and tested them in the lab. Those are the ones that work in our physiological and cognitive studies. So it doesn't mean that there may not be other ones out there. We have several devices from several companies that are waiting in line for us to test. And the other one is Cell Gen Therapeutics.
Yes.
Cell like in cells, Gen like GEN, and therapeutics. And that one is in Dallas. They make this so-called HD laser, high density laser, that we have used as the first one that we use. And the ones in Cyton Sys were the ones that they were tailor made to our specifications. And now he's commercialized this later on.
So that's our episode. I hope it was useful to you. Check out the show notes on iTunes or on our website where we've summarized the key points. If you find the information here valuable, please consider giving us a 5-star rating on iTunes and leaving comments in the comments section. It will really help us bring this message to as many people as we can. If you have questions or comments, connect with us on social media. Finally, to support us, go to patreon.com/evolvingpast and consider hitting the button and becoming a patron of this show. That's Patreon, Patreon, patreon.com/evolvingpast. Thanks, and talk to you next time.